Hello, everyone, and welcome to our webinar series titled To Immunity and Beyond, focusing on immunity and infection in donation and transplantation. My name is Erica, and I will be today's moderator. Firstly, I'd like to acknowledge that the CDTRP is hosted by the University of Alberta, which is located on the Treaty 6 territory, a traditional gathering place for the diverse Indigenous peoples, including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Saluto, Anishinaabe, Inuits, and many others whose histories, languages, and cultures continue to influence our vibrant communities. I encourage you to write your land acknowledgement in the chat box. We'd also like to acknowledge CIHR, whose generous support enabled us to host these sessions. Today's session is recorded and will be posted on the CDTRP website and YouTube channel. During the presentations, please keep your mic off. If you do have a question or comment, you can raise your hand with the raise hand button or just type it into the comment section. Today, we're pleased to have Dr. Anna Konvalinka present her research titled Predicting Kidney Allograft Outcomes and Potential Treatments Using Protein Signatures. Dr. Konvalinka is a clinician, scientist, and transplant nephrologist at the University Health Network, an assistant professor at the University of Toronto, and a senior scientist at the Toronto General Hospital Research Institute. Her research centers on antibody-mediated rejection and kidney allograft fibrosis, using systems biology and proteinomics to uncover mechanisms, identify biomarkers, and explore drug repurposing for kidney diseases. She is also the multi-organ transplant biobank. She's also directs it at the Ajmera Transplant Center and has received multiple national and international awards for her contributions to transplant research. Speaking for myself and hopefully many others, I'm very excited about your presentation. I will now hand it over to you.
Thank you so much, Erica, for the invitation and for this very kind introduction. So I'm going to share the slide. And I hope that everyone can see the slides, right?
Yeah, looks great.
Okay, great. Let me just minimize your faces so I can focus on the presentation. Okay, so Erica has already introduced me. The title, I had forgotten which title I had given you, but that's okay. The topic is still the same. And so I'm going to tell you about one of the research themes that we're working on in the lab. So this is the overview of my talk, and I'll first begin with the background. So kidney transplantation is the best treatment for patients with end-stage kidney disease. However, most grafts fail prematurely. An antibody-mediated rejection, or AMR, is the leading cause of premature graft failure and accounts for more than 50% of late graft losses. And unfortunately, the pathophysiology of antibody-mediated rejection is incompletely understood, and there is a paucity of effective treatments. And so this is something that we think about in the lab a lot. And so when I think about the pathophysiology of antibody-mediated rejection, I think about the injury that takes place in the microcapillaries of the kidney. And there within the capillary network, we can see the endothelial cell that sits on the basement membrane and constitutively expresses human leukocyte antigen class 1 molecules and frequently expresses human leukocyte antigen class 2 molecules. If these particular molecules are different between the donor and the recipient can develop donor-specific antibodies or DSA against either class 1 or class 2 HLA molecules. And it's generally accepted that these can cause injury directly to the endothelial cell through complement activation, which we see as complement C4d fragment positivity in the peritubular capillaries on the biopsy. And lastly, through antibody-dependent cell-mediated cytotoxicity, whereby the Fc domain on the antibody ligates Fc receptors on natural killer and myeloid cells, resulting in cytotoxicity as well as the release of inflammatory cytokines. And a particularly challenging and mysterious form of antibody-mediated rejection is transplant glomerulopathy or chronic glomerulopathy, which entails remodeling of the basement membranes. And there's a conundrum in transplantation in that 30 to 60% of patients who do develop donor-specific antibodies after transplant or before transplant do not develop rejection. And so we wondered how these antibodies may be different. And we also wondered if there were any tissue adaptations to donor-specific antibodies that protected certain grafts from DSA. So when we talk about these donor-specific antibodies, we talk mainly about the binding specificity of the antibodies that is determined by the Fab domain, and that's what the HLA lab examines. However, it's really the functional interactions with the Fc receptors that is determined by the Fc domain that is critically important in the function of an antibody. So humans have 5 activating Fc gamma receptors and a single inhibitory Fc receptor 2b. And these are expressed on a variety of immune cells. There's another interesting thing about this Fc domain, and that's that a single asparagine residue is very heavily glycosylated. And these glycosylations determine the function of the Fc domain. For example, sialic acid activates inhibitory Fc gamma receptor 2b on monocytes, macrophages, or B cells. Lack of fucose can activate Fc gamma receptor 3a on NK cells or myeloid cells and result in cytotoxicity. And galactose can promote complement activation. But these, we do not monitor these glycan modifications in the clinic. So I'm going to tell you first story, and that's about kidney tissue interactions with DSA. And so we decided to study these phenomena of DSA positivity and very different outcomes in our kidney transplant recipients. So we wanted to recruit patients who all had a DSA after kidney transplant and who had a concomitant kidney allograft biopsy. That could be classified into those who had rejection and those who had no rejection. And in parallel, they had a blood sample that showed the donor-specific antibody. And there we wanted to examine these modifications on the antibody to see if they at least correlated, and subsequently to study further mechanistically if they contributed to activation of various Fc receptors or complement. And so I'll first focus on the kidney tissue proteome or kidney functional interactions with DSA. And so this is the work that was led by, and is still led by Dr. Kieran Mannion in my lab, who is a scientific associate and who used the same method that we had published on previously in JSON in 2020. So Kieran was able to identify 116 patients, all positive for DSA in our transplant center at UHN. And all of these patients had kidney allograft biopsy after transplant, for-cause biopsy, and had tissue stored in pathology. And the patients could be classified as having no rejection, having active antibody-mediated rejection, chronic active antibody-mediated rejection, or mixed cellular and antibody-mediated rejection. Kieran then went on to use laser capture and microdissection. So I'll show you how she could use laser to dissect and analyze glomeruli and tubular interstitium separately. And so this is what kidney biopsy looks like under this dissecting microscope. And so she can use a laser, and just like I did here with a pen, she can circle and cut out glomeruli and then separately tubular interstitium. And we normalize this by surface area or volume of the tissue that was being collected to take the exact same amount per biopsy. And then Kieran examined this tissue separately on using mass spectrometry by Q-exactive mass spectrometer to determine all the different proteins that were in each of these different compartments in every biopsy. And then we performed some follow-up analysis. So to show you some patient characteristics, these are the 4 patient cohorts here. Most of the patients were male in all 4 groups. Because these were for-cause biopsies, they were done at different time post-transplant. You can see that patients with chronic active antibody-mediated rejection tended to be biopsied further away from transplant. And you can see that, unfortunately, a substantial proportion of patients with chronic active or mixed rejection experienced graft loss during the follow-up, and there was a median follow-up of about 5.9 years from the biopsy. In addition, I use this heat map to show you the various donor-specific antibodies that the patients had. You can see that patients with no rejection or with active antibody-mediated rejection tended to have one donor-specific antibody against either class 1 or class 2 antigens. Whereas patients with chronic active or mixed rejection tended to have more than one antibody and predominantly class 2 antibodies like anti-DQ. And so this heat map illustrates their BANF or pathology scores that the pathologists use to assign certain diagnoses. So patients with no rejection did not have any evidence of significant inflammation, either microvascular or interstitial, but they did have some interstitial fibrosis and tubular atrophy and in varying degrees of hyalinosis and intimal thickening. Patients with active antibody-mediated rejection did not have any chronic glomerulopathy by definition, and all patients with chronic active antibody-mediated rejection had chronic glomerulopathy. You can see that patients with mixed rejection, very few of them had evidence of chronic glomerulopathy, but almost all of them had C4d positivity, and only about half to a third of patients with active and chronic active antibody-mediated rejection had C4d positivity. So Kieran then went on to analyze these proteomes from the biopsies. And so she was able to identify and quantify over 2,500 proteins in the tubular interstitium and over 1,700 proteins in the glomerulus. And then using very stringent filtering, she focused in on 389 proteins in the tubular interstitium and 127 in the glomeruli that were significantly differentially expressed between any of the 4 groups of patients. And so I will first show you what she found in the glomerulus. So she used unsupervised clustering analysis, and you can see here on top these 127 proteins significantly differentially expressed in the glomerulus. Here you can see first chronic active, mixed, and no rejection. Red color indicates increased relative expression of the protein, and turquoise color indicates decreased relative expression. And you can see that certain clusters of proteins with similar expression were identified. And we became interested in these 2 clusters in red rectangles because they contained early components of the complement like C3, CFH, C4a, but also late complement components like C9. And these proteins were all significantly increased in chronic active antibody-mediated rejection in the glomeruli compared to the other groups. So we then did a similar analysis for the tubular interstitium. And here in the clustering analysis, Kieran again identified a group of proteins that contained all the proteins involved in the terminal complement cascade or the membrane attack complex from C5 to C9, including the regulatory proteins like vitronectin and clusterin. And all of these proteins were significantly increased in chronic active antibody-mediated rejection compared to the other groups. And so we then wanted to know if these complement proteins actually correlated with clinical and histopathologic characteristics. And so first focusing on the glomerulus, this is a bubble plot which shows you wherever you have a circle, significant correlation.
Laser.
Sorry? Oh, significant correlation between one of the features displayed here on the y-axis. And you can see that early complement as well as late complement correlated significantly with chronic glomerulopathy. You can see that C9 late complement was co-clustering with IgM, but not IgG. And that IgM as well correlated significantly with both glomerulitis and chronic glomerulopathy. In contrast, in the tubular interstitium, we saw that this entire group of membrane attack complex proteins correlated significantly and directly with interstitial fibrosis and tubular atrophy. And so with this in mind, we next wonder whether the expression of these proteins correlated with C4d positivity in the same biopsies. And as you can see here, we plotted glomerular proteins and tubular interstitial separately. And with the exception of C3, which was higher, which had correlated with higher C4d positivity, in the glomerulus only, there was really no other correlation between the expression of these complement proteins and C4d positivity in the biopsies, suggesting that C4d positivity may not be telling us the whole story about complement. So in addition to looking at these correlations between BAMF or pathologic scores and the proteome, wanted to know if we could use the entire AMR proteome to predict who is going to develop graft loss. And to do that, we had some help from Dr. Jishnu Das from the University of Pittsburgh, who is a computational biologist, and his student Augusta Vincent. And they decided to use an interesting machine learning method that they recently described, which is called SLIDE. And this machine learning method is interesting because for any given outcome, for example, in this case, graft loss or no graft loss, the method is able to identify not only observable features like the proteins in our biopsies that correlate with the outcome, but can also identify these latent or hidden factors in the data that explain or underpin the outcome. So we got interested in this and wanted to see where our data would take us. So we gave them the entire AMR proteome separately for glomeruli and tubular interstitium, and I'm only going to show you the glomerular compartment in the interest of time. And so Augusta was able to identify 3 latent factors here, Z4, Z6, and Z100. And I'm showing you the proteins in each latent factor that correlated with the latent factor. You can see that all 3 of these latent factors contained complement proteins, which were increased in orange in patients who ended up losing their graft during the follow-up, whereas vitronectin, the negative regulator of complement, was significantly decreased in patients who went on to lose their grafts. And so furthermore, we generated a composite SLIDE score from these 3 latent factors. And based on this SLIDE score, we were able to distinguish patients with graft loss from those who experienced no graft loss during the follow-up. And we were able to do so accurately even when we split the biopsies into active antibody-mediated rejection, chronic active or mixed rejection, even though we didn't provide Dr. Das any information about the underlying pathology or BAMF scores. Furthermore, this composite SLIDE score correlated significantly and directly with serum creatinine, a measure of kidney function, with the number of class 2 donor-specific antibodies, and with the degree of this chronic glomerulopathy. And lastly, from these 3 latent factors, we were able to identify 2 proteins, C3, complement factor 3, and integrin alpha 3, another important podocyte protein, which alone were able to significantly distinguish biopsies from patients who were going to go on and lose their graft compared to those who would not. And so, of course, complement was of interest to us. And so our next question was, where is the complement actually expressed or deposited in the kidney? And this is ongoing work. This is, I'm going to show you some preliminary work from Maya Allen, who is a PhD student in my lab and who used spatial proteomics method called imaging mass cytometry. The power of this method is that it is able to combine up to 40 different antibodies of interest, each targeting a protein of interest on a single slide. And each of these antibodies is tagged to a heavy metal tag. So when a laser is shot in a specific index location, these metal tags are released, providing information both about the quantity of the protein being labeled and its location. And so Maya first wanted to look at C3, given how important C3 was in the glomerulus. And so you can see here an image of a living donor pre-implantation normal biopsy. And you can see that C3 is virtually absent from here. However, in a patient with chronic active antibody-mediated rejection, you can see that C3 is predominantly increased and predominantly expressed in the glomerulus. And you can see that based on the marker CD45, C3 does not coincide at all with the immune cells, which are mainly in the tubular interstitium, but not in the glomerulus. However, C3 coincided almost entirely with IgM, but not with IgG, which is interesting to us because the current paradigm is that it is IgG-based donor-specific antibodies that are doing all the damage and that we monitor. We don't even monitor IgM. And similarly, Maya wanted to look at the terminal complement cascade. So she examined C5a and C5b9. And as you can see in a healthy living donor biopsy, these were virtually absent. However, C5a in a patient with chronic active antibody-mediated rejection colocalized mainly with the collagen 4, which is in the basement membranes, both in the glomerulus and the tubular interstitium. And C5b9 was predominantly identified in the peritubular capillaries, and there it coincided with IgG. And so this was interesting to us. And so to conclude this first part, I've shown you that unbiased proteomic analysis uncovered significantly increased complement, both in the glomeruli and in the tubular interstitium in antibody-mediated rejection. Early and late complement components in glomeruli with antibody-mediated rejection were predictive of graft loss during follow-up. And C5a anaphylatoxin is mainly deposited in basement membranes, while terminal complement is activated in microvasculature of chronic active antibody-mediated rejection and colocalizes with IgG. So with this, I'm going to switch gears and now tell you about donor-specific antibodies and their glycans. So now I'm going to talk about the right side of this slide. And so our next bit, which was asking the question of, are there any modifications on these donor-specific antibodies or any other properties about these antibodies that could explain why some patients do not develop rejection and others go on to develop rejection after development of these antibodies? This is the work again with Dr. Jishnu Das and with Dr. Anirudh Sarkar from Georgia Institute of Technology, who is a biomedical engineer who is able from just 5 microliters of blood to isolate anti-HLA antibodies using single antigen beads, just as we would do in the clinic. And then for each antibody specificity, using labeled probes, he can identify the isotype or subclass of that antibody and determine the binding of that antibody to Fc receptors and C1q, as well as using lectins, he's able to decipher if there are specific galactose, fucose, or sialic acid modifications on these antibodies. Of course, this method generates a lot of data, and the data is analyzed further by Dr. Das and his team. So our first question was whether DSA and their modifications can predict rejection. And so this project was led by Chirupa Chakraborty, who is a PhD student in Dr. Das's lab. And she recruited 36 patients with a kidney transplant from Pittsburgh who were all positive for DSA, half of whom had antibody-mediated rejection, and the other half who had no rejection. And we also recruited a similar number of patients from Toronto who fit the same criteria. Chirupa then used SLIDE, and her main question was whether we can identify any latent factors in these antibody modification data or the types of antibody data that could predict who's going to develop antibody-mediated rejection or will have no rejection. And if you're interested, this paper is currently in preprint and in revision in Kidney International. The first observation that Chirupa made was that those antibodies that were detected early, within 3 months post-transplant, were very different from those that were detected late. The early antibodies in the first 3 months post-transplant are most likely to be memory responses. And so she separately analyzed these two types of data. And so for the early antibodies, you can see here area under the curve for prediction, for accuracy of prediction. So she was almost 100% accurate in predicting who is going to develop antibody-mediated rejection or stay stable without rejection in this group of Pittsburgh patients who all had donor-specific antibodies. And then she also validated the accuracy of this prediction in the Toronto cohort. You can see with 75% accuracy. And so she was able to identify two latent factors that I'm showing you here, which were responsible for the accuracy of prediction. And you can see these factors contain IgG3, 1, IgM, and SNA, which stands for sialic acid. So it follows that we can accurately predict who has antibody-mediated rejection or no rejection in these positive patients based on serum IgG, IgM, and sialylation. Chirupa also wanted to see if we can identify if patient-specific, donor-specific antibodies were enriched in some of these latent factors. And you can see that IgG3 that targets donor class 1, specifically this A68, was higher in the rejectors and enriched in latent factors. And similarly, that IgG1 that targets class 2 HLA is enriched in latent factors and also elevated in the rejectors. Chirupa then turned her attention to late rejection. And so here as well, she was able to develop a model that accurately predicted who is going to develop AMR and who will not. And she validated this prediction model in the Toronto cohort. This time, however, she identified 5 latent factors that were responsible for the prediction. And you can see that they consisted of IgM, sialic acid, IgG, and C1q binding. And here, interestingly, the probes that were particularly increased in rejectors and enriched in these latent factors included IgM antibodies against class 1, as well as IgG against class 2 antigens. Again, changing around our paradigm of IgM, which we don't even monitor in the clinic. Lastly, Chirupa asked what differentiates early from late antibody-mediated rejection. And so here she again used slide to develop a predictive model, which was accurate and much better than this permuted prediction. And again, she was able to validate it in Toronto cohort. And here you can see 2 latent factors that were responsible for the prediction. One that contained galactosylated antibodies and the other that contained IgM. So it follows that galactosylated antibodies against class 1 and 2 HLA and IgM against class 1 HLA are enriched in latent factors that differentiate late from early antibody-mediated rejection. And it was interesting, this galactosylation was particularly interesting because we previously, we identified this older paper that showed that FC galactosylation promotes hexamerization of IgG1 that leads to enhanced classical complement activation, linking this galactosylation to the complement that we're finding in the tissue. So to conclude this part, in case of de novo donor-specific antibodies, IgM against HLA class 1 and IgG against HLA class 2 can predict antibody-mediated rejection. And IgM donor-specific antibody differentiates patients with late from those with early antibody-mediated rejection. This is consistent both in the blood of the patients and supported by tissue proteomics in the biopsies. Lastly, galactosylation of donor-specific antibodies differentiates patients with late from those with early antibody-mediated rejection, suggesting distinct ability to activate complement. And so I'm going to show you now where we're going with this. So I've shown you that we've identified both early and late complement components as being significantly differentially expressed in the 2 different compartments of kidney biopsies with antibody-mediated rejection. We want to understand next, first in detail, what is the source of complement in the kidney antibody-mediated rejection, whether it's locally produced or deposited, and if it's locally produced, which cells produce it. In parallel, we will examine complement activation in blood and urine. And lastly, we're working to develop a model to study complement inhibition ex vivo, and a model to use patient-specific sera to simulate this complement deposition on endothelial cells in culture. And as well, we have this entire cohort that had biopsies that were analyzed by my group, also had blood collected. And so we're further analyzing these modifications from the blood of these patients in order to put these two large sets of data together in order to build potential testable mechanisms of injury that we can subsequently evaluate in vitro and in vivo in preclinical models. And with that, I will thank the people in my lab who do this work. And I will thank my collaborators working on this project and sources of funding. We're now funded through CIHR to continue these studies. And I would be thrilled to answer any questions.
Thank you for that excellent presentation. I am going to open the floor for questions. I'm going to take the opportunity to ask the first one.
Go ahead.
Early on, very early, you mentioned you have 116 patients. You mentioned that the sex was higher in males. The sample size, there was a lot more males. Is there a sex difference for graft loss between males and females?
Great question. Well, there is a sex difference in a sense that we mostly, most of our patients are males. We transplant males more. There are more males in the groups. In this particular cohort where, all the patients either had no rejection or had a form of antibody-mediated rejection, we did not see a difference between male and female recipients who did develop one of those in terms of graft loss. In general, what we know is from prior literature, not my own, is that there is a bit of a discrepancy. First of all, females, because they can get pregnant, they are more likely to be sensitized, so to have antibodies against others in the population. And so they may be harder to transplant. And similarly, and this is an interesting observation, when females get a kidney from a male, they're more likely to reject that kidney sooner than males are if they get kidneys either from other males or from females. And, this is not fully understood. Some of it is thought to be an immune response against the proteins that are encoded by the Y chromosome genes. But it's more complicated than that, and probably there are some sex hormones playing a role.
Thank you.
Hope that answers.
Yes, it does.
I
Ian?
have a question.
Dr. Rogers?
Great talk. Super interesting. I have a very basic question. How does T-cell mediated rejection fit into this? Does it interact? Could it impact the results, or is antibody very separate entity and almost mutually exclusive?
Yeah, great question. I think the current paradigm is that rejection is one big continuum. So it's basically all part of the immune response. It's just antibody-mediated rejection is at one end of the spectrum, where the immune system has done everything already once it develops the antibody. But it's the same spectrum.
Okay.
So, you did notice accurately that we have a subset of patients who had mixed rejection, so both cellular and antibody-mediated rejection. Yeah. And they do have, particularly interesting changes in the tubular interstitium. So when we look at the differences in the tubular interstitium, I've mainly focused on the glomerulus, but, they're driven by this group of patients. And so we know that these patients tend to have more interstitial inflammation and more tubulitis, so more infiltration of the immune cells beyond the basement membrane, so breaking the basement membrane in the tubules. And so and here, even in the proteome, we do see the impact of that. And, the impact is such that we see a lot of metabolic change in the tubular intestinal compartment. So there is, there's something, we didn't include the group with cellular rejection alone because we were mainly focused on donor-specific antibodies and those differences. But when we do include them, because we have a small subset of patients with cellular rejection, they do look different. They're not quite the same as the ones that have mixed rejection. So,
thank you.
No problem.
We have a question in the chat by Dr. Cornell. Can you tell us more about the glomerular staining for IgM and C3? For example, what is the staining pattern? Normally by immunofluorescence, glomeruli staining for IgM and often IgM, mostly mesenchymal, and is there more staining in the settings of proteinuria and transplant glomerulopathy? Can you distinguish this normal staining from what I think you are suggesting, and there may be IgM DSA in the glomeruli? Sorry about that.
Yeah, great question. And I mean, I think I'm still working to kind of figure out, because these are very preliminary data. We now have a bigger cohort that we're going through the analysis with. And, I'd be interested in, I'm going to reach out to you separately, actually. But I would be, one thing that I was looking to see in literature is whether there is a center that does immunofluorescence on all comer biopsies, because our pathology department only does immunofluorescence when we report that there is significant proteinuria or we're questioning recurrent disease. I mean, they do IF for C4D, but not for the immunoglobulins. So I don't have a sense for, whether we're identifying immunoglobulins at all in the glomeruli, and so on. But anyway, so I don't know what happens like in all comer biopsies. So what we are seeing here is we don't see any deposits in kind of our no rejection control that are DSA positive, and in these living donors that I showed you. So we don't see any deposition in the glomeruli of either IgG or IgM. You're asking me whether the deposition is mostly mesangial. Probably, because when we don't have a good mesangial cell marker, unfortunately. So we didn't include it in the IMC panel, but we have endothelial cell markers. We have glomerular basement membrane, and we have podocyte marker nestin. And I don't see, in most cases, not all, but in most cases, I don't see a clear colocalization of either IgG or IgM with CD31 or with nestin or with the basement membrane, collagen 4. So, so it's likely that it is in the mesangium in these cases, but I think we have to systematically examine all the regions of interest for all the biopsies that we'll be studying to answer that, fully.
All right. Dr. Cornell would love to talk with you after this talk, so if you want, I can set that up between the two of you. So
great.
Here she says, thank you. That's a full answer. I'll speak on her behalf. I have a question kind of about if you could explain the difference or relationships. Throughout your talk, you mentioned early, late complementary proteins. You say terminal complementary proteins. You say latent factors. What's the relationship and difference between these three or these four?
Yeah. So basically, I wish I had put in my scheme of here we can share maybe again this scheme. Let me just try and share again my slide just to illustrate the point better, because picture is worth 1,000 words. So for example, in this image, what you can see by early complement components are C3, also C4 and C2, which are even above upstream of C3, which are initiating injury through either classical or lectin pathway. So those ones I would consider the early complement components, whereas following the formation of C5 convertase that cleaves C5, Those would be the late complement products. And in this case, C5a anaphylactoxin can be released, which can actually stimulate and activate T cell, for example, but can also stimulate and activate myeloid cells. While C5b-9 can get deposited like this in the membrane and form the membrane attack complex. And so basically when I say terminal complement, I mean that's kind of similar to saying membrane attack complex proteins. So basically it's just that we were predominantly seeing the earlier complement in the glomeruli, and in the tubular interstitium, predominantly the late or terminal complement. When I say latent factor, so that is simply a term derived from this slide machine learning model. And so latent factor means that this is a sort of a hidden factor in the data that links the observations to the outcome. So proposing that there's some anchor proteins in the data that actually can explain the link between all the proteins in the proteome that we're identifying and quantifying and the outcome being graft loss or no graft loss. So, so when I showed you the latent factors, I showed you all the proteins in each latent factor that were kind of in these anchoring proteins or most important proteins that kind of explained this relationship. Hope that makes sense.
Yeah, very clear now. I'm going to go to Sean Delaney.
Hi, Anna. Great talk.
Hi.
I was going to take a slightly different one because you have a fascinating group of people on here. Many of you know I've worked in this field for 20 years and I'm a two-time transplant recipient, but over probably 24 or more research studies I've been part of. This has been some of the most interesting work I've done, which is being a patient partner advisor with Anna's team. And because it's working in basic sciences, because usually when someone calls up and say, hey, do you want to be part of a research study, it's for a drug trial, or it's for a quality of life trial, or a vaccination trial, or something very easily understood. And I congratulate Anna and her team for including Sadia and myself. And I would just like to hear a little bit more about your experience, and also for the sake of others out there doing basic science research and how that's worked, because basic science is not the easiest thing to explain to the general public. And Sadia and I have been drinking from the fire hose for 2 years.
Thank you so much for that comment, Sean. I've really enjoyed meeting you and sort of, involving you in this project. I'm glad that Joe Gallagher is also online. And so he's my next patient as well, partner on the new CHR grant. So I'm hoping to engage him very shortly with this project as well. Nice. So I think I'm glad to hear that it was interesting to you, and that you find it promising, and that you got something out of it. For my trainees in particular, and for the other trainees like that I was mentioning from Dr. Dassi's lab and Dr. Sarkar's lab, I mean, I think it's a really unique and enriching experience to meet you guys and to hear your side of the story and your lived experience, because this is what motivates them. Then they go back and they can see, there's a light in their eyes. They can see why we're doing what we're doing. They can understand why we're asking this question, and they have it now kind of attached to a specific person. It's very different because they're not MDs, so they're graduate students in the lab. So it really motivates them. In fact, like my 2 PhD students now want to be clinician scientists, so they want to connect the two, and this is how inspiring that's been for them. So one thing I will say, I was just at the ATC in Boston, American Transplant Congress in Boston, and there was, because of all the political landscape that we're facing now, there was a question of how do we kind of pull together, and how do we get funding, do we get funding from the government and all that. And I think patient partners as well, we have a lot to say and could kind of lead some massive change by, appealing to the government and through various organizations, kind of emphasizing how important it is to do the translational and basic science research, because this is something that I think the world is forgetting now. They're forgetting how we got here. We got here through translational and basic science.
100%.
Yeah, so I think if you can come up with a way, Sean, that we can harness the power and the potential of people like yourself who are really motivated to see this, and I think I would stand behind you 100% and help you in any way I can. But I think this is kind of where we should probably put some efforts in the future.
I mean, even our transplant labs doing standard HLA testing and things like that are fly under the radar, and I think basic science flies even further, even for those of us as patients and family members. And I think it is important that we at least understand more. And so I absolutely appreciate you've done this, and I think for others, this is a plug for CDTRP. But I would encourage you, if you are interested at all in engaging patients, contact Manuel from the CDTRP team. And that's how we were connected up with Anna's amazing work and team.
I agree. Manuel is fantastic at what he does, and yes, that's if you want to connect with us, he's the person to do so. Thank you for sharing that, Sean. We have another question in the chat from Dr. Andre. Sorry, I got distracted. Did you think this distinct DSA presentation could suggest tolerance, reflecting the patient's propensity to produce less pathogenic antibodies, particularly in those with long-term graft survival?
It's an interesting question. I mean, I think one of our hypotheses was that it maybe I would call it accommodation, when we started to work on this. So we were looking for signals of accommodation in the tissue, and this is a very poorly described phenomenon, but in the way that it had been described before. So we gathered a list of all the genes previously evaluated in the context of accommodation, and we screened to see whether we're finding the same proteins and if they're increased in patients who have donor-specific antibodies but no rejection. We did not find such a signal. So there was in our case no signal of accommodation. What we did find was that there was a mostly metabolic and kind of proximal tubule phenotype specific program that seemed to be enhanced in those patients who went on to maintain their grafts and who had no injury versus those who had severe injury. So, it seems that there's some metabolic, that it's more like it's not, well, I don't know yet the antibodiome data, so I can't comment on antibody modifications and or specific antibodies that are going to play a role here. But I do think that it has to do mainly with this kind of acute to chronic kidney transition and injury, and probably some metabolic adaptations of the tubular interstitium and tubular cells specifically.
I'm sure he says thank you. Yes, perfect. There it is. Thank you. Mr. Gallagher, you're up.
Oh, that was great. That was a title. I don't know. That would be a great title to have. Quick question. You had mentioned earlier in the presentation about being surprised to find IgM antibodies being a main driver as opposed to the IgG. Just wondering if that's going to drive any clinical practice changes, and also are there currently any therapeutic agents to sort of address that, or things in the pipe?
Great question, Joe. I'm so glad, and you're going to be part of this, so you're going to help us answer all these questions. So I mean, the goal is ultimately to translate this to patients. I wouldn't be doing any of this if that was not my goal. But I can't call them yet causal, and I don't know if this is a spurious observation in one cohort. What we are doing, and also we want to mechanistically test these antibodies. So we want to elute these specific IgM antibodies and see if they're able to activate complement in the tissue. So in our, we have a setup with the same cells, the endothelial cells that line the microvasculature of the kidney. We have the same setup. And so we want to test and see if, we can demonstrate that these antibodies activate complement, or how they bind the FC receptors, if there are any differences. And if the answer is yes, then I think, in a similar way in which, Anne Halpin and Lori West, for example, are developing ABO-incompatible assays on a single antigen B, just like the HLA assays exist now. We could do the same, just use IgM. That would be even simpler, actually. So, so I think it would be relatively, in my mind, relatively simple to translate that into the clinic, at least in terms of the monitoring. Now, if we understood better who had this IgM and what it was doing, then we could come up with better treatments.
Okay. Do we have any last questions? I see that we're almost at time. All right. So, Dr. Konvalinka, on behalf of CDTRP, thank you so much for presenting. If anybody has any questions, comments, or suggested topics and speakers, you can contact myself at ekcroft@cdtrp.ca. I want to thank everybody for joining. We do hope that you're going to have a great summer, because we're going to see you back on September 3rd. We are taking a summer break. So, all of you, enjoy the rest of your day. Take care.
Thank you. Bye.